Podcast. I'm your host, Nick Jikomis, and today I'm speaking with Dr. Maayan Levy. Maayan is an assistant professor of microbiology at the University of Pennsylvania, where her lab studies the communication between the microbiome and its host, with a focus on the gut microbiome and its relationship to diet, immune system function, and disease. We discussed a variety of topics, including her recent study linking the ketogenic diet to lower risk of colon cancer, what the ketogenic diet is, and what the metabolic state of ketosis does in the body, molecules called ketone bodies, such as BHB, and the extent to which BHB supplementation can recapitulate some of the physiological effects of the ketogenic diet, how the microbiome influences the function and development of the immune system, and the hygiene hypothesis, the idea that modern human cultures may be too clean or too heavily reliant on antibiotics and other types of technology that actually interfere with our microbiome in ways that may be detrimental to metabolic and immunological health. If you're interested in health and diet, generally speaking, and in the microbiome and the immune system in particular, this is a very interesting episode. I did not know too much about the ketogenic diet or the metabolic states it induces and how those can be linked to different disease states and things like that. So if you're into any of that stuff, I think this is a really fascinating topic with a researcher who has just recently put out a lot of new work looking at this, new scientific work. So this is really cutting edge stuff that we discussed. As always, if you enjoy the content of this podcast, please like, share, and subscribe. You can share your favorite episodes with friends and family. You can also sign up for my free weekly Mind and Matter newsletter at mindandmatter.substack.com. I also post some other long-form content there from time to time based on the topics covered on the show. And you can find all of the different versions of the podcast, links to the audio and video versions and different things at that Substack. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things, and there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. Today's show is brought to you in part by Dosist, an all-natural cannabis company specializing in dose-controlled cannabis products made with plant-based ingredients. To learn more about Dosist, their products, and where they are available, please visit their website through the link in the episode description. And with that, here's my conversation with Dr. Maayan Levy. Dr. Maayan Levy, thank you for joining me.
Thank you for having me.
Can you start off by just telling everyone who you are and what your lab works on, what kind of science that you do?
Sure. So I am an Israeli scientist. I was born in Israel and I was trained in Israel. I did my master's and my PhD at the Weizmann Institute of Science in the lab of Eran Elinav, studying the microbiome. I came to the University of Pennsylvania in 2018. And I've opened my lab in the microbiology department. And generally, my lab is interested in studying questions that are related to the microbiome, intestinal immunity, how the communication between the gut and the bacteria is happening. So we are mainly focused in the gut and how this kind of interaction is influencing different human diseases.
And how did you get interested in that general subject? Were you always studying this, or were you studying something else and then kind of got into this?
So I think that, about like 10 years ago, there was a major focus and a shift into the microbiome. And I think this mainly happened because, we understood that our genomes cannot explain everything. There are diseases and different conditions that cannot be explained by alterations of our genome. So there must be something else, and this something else is our microbiome. And if you are thinking about the gut, then the number of genes that are contributed by bacteria are much larger than the amount of genes that are contributed by us, by the host. So the microbiome has been over these past years shown to be relevant in basically any aspects of human physiology and human disease. So now you can connect the microbiome to almost any disease, from neurological diseases, cancer, aging, different metabolic diseases, lung diseases, and like basically everything. So we know that the microbiome is super important for many, many aspects of our lives.
Yeah, I mean, that makes a lot of sense. Just, when you think about how numerous these microbes are in terms of the number of cells and the number of genes, it really does make a lot of sense. And
it's not just the number, it's also how close they are. So if you're thinking about the gut, the amount, this huge amount of bacteria that we all have inside of us is located so close to what we know and need to maintain as sterile tissue. So they're so close. So we are sensing them constantly. We have to constantly react and respond to their presence. But on the other hand, we need to make sure that we don't constantly have an inflammatory condition and a constant response towards these bacteria.
Yeah, so that means, it's very interesting. On the one hand, you have to have this sort of exquisite sensory system to constantly detect and identify these microbes, and yet it somehow has to discern that they are not bad or not always bad, so that we don't have this constant sort of immune reaction.
Yeah, exactly. And so I think that the way I see it, the major cell type that is mediating this protection or sensing is the intestinal epithelial cells. If we are focusing on the gut, obviously there are microbes outside of the gut, basically everywhere that our body is exposed to the environment, the skin, the lung, the reproductive tract, but my focus is in the gut, so I can say that the cell type that I think mediates this interaction is intestinal epithelial cells. These are the cells that line the gut. So they are the cells that see the microbes and what they produce first. And if you're thinking about sensing mechanisms, by now we know that there are several types and several families of receptors that can sense the presence of bacteria. And during my PhD, I actually worked on one of these sensing platforms that is called the inflammasome. And I worked specifically on an inflammasome that is called NLRP6. It is called NLRP6 because the sensing component to it is an NLR protein that is called NLRP6. So we call it the NLRP6 inflammasome. And this was discovered back in 2012 by my PhD mentor, Dr. Ran Elinav, and what he found is that this specific inflammasome is expressed in epithelial cells in the gut, and that in the absence of this sensing platform, the mice develop an abnormal microbiome composition or a dysbiotic microbial composition. And more interestingly, what he found is that this microbiome made the mice more susceptible to different inflammatory diseases, IBD, but also diabetes, and even colorectal cancer.
I see. So if you
knock it out, if there is no sensing, the microbiome becomes abnormal. The composition is not what it used to be. It doesn't look like a healthy composition, and the function of bacteria, more importantly, is altered as well.
I see. So if you disrupt the ability of the cells lining the gut to actually sense the bacteria that are present, they presumably can't figure out which bacteria are which.
Exactly.
And the general consequence of doing that, if you completely disrupt it, is the microbiome composition is different, and you're more susceptible, at least rodents are more susceptible to all kinds of different diseases.
Sure. Yeah, that's exactly this. And so this was basically the initial observation. What we then did in subsequent studies to show exactly how it happens. We found that the microbes can influence the activity state of this sensing platform. And downstream to the sensing platform, there is secretion of inflammatory cytokines, IL-1 beta and IL-18. And we found that downstream to these cytokines, there is a transcriptional program inside intestinal epithelial cells that leads to a secretion of antimicrobial peptides. And when you disrupt this, you disrupt the secretion of these small peptides that are antimicrobials. And if you don't have these antimicrobials, then the community of bacteria goes wild.
I see. So there are systems that these cells are connected to, which probably regulates the type of natural antibiotics, basically, that our body makes.
Exactly.
And how much we're making. And if you disrupt that, you make too much or too little or the wrong ones, it just screws up what kind of bacteria are actually present.
Exactly. So you shift the balance somehow when it becomes abnormal and more likely to develop different intestinal diseases.
And to what extent? So when we think about the body's internal ability to sense and identify bacteria, how much of this are we born with? Like how much of the knowledge is innate and how much of it has to be learned as development is happening?
So we are mainly focused on the innate immune system that we are born with. I don't think it needs to be trained. But if you're thinking about the coevolution of bacteria and the human host or mice, then we are born sterile and then we acquire the bacteria. So as far as I know, there is no activation of this sensing system before we are born when we are sterile. And then after we are born and we acquire the first bugs, then we start sensing and reacting and maybe learning what we need to respond to and what is a friend.
I see. And so there's this distinction between the innate and the adaptive immune system that gets made in immunology. Can you just sort of explain what that is for people at a very basic level?
Yeah. So the immune system is divided into two main arms, the adaptive immune system and the innate immune system. When we're talking about the innate immune system, we talk about receptors that can detect different molecules, and these receptors, we all have the same ones. They don't undergo any recombination, in contrast to receptors of adaptive immune cells, mainly B and T cells. So what we are born with is what we have. There is no generally, it's thought that the innate immune system doesn't have any memory capacity, in contrast to an adaptive immune system or an adaptive response. If you challenge with a pathogen that is, then the adaptive immune system will respond. And then in a secondary challenge, the response will be even larger or more robust. This is something that doesn't happen when something is sensed by the innate arm. So it's a more basic, maybe, or more primitive.
The responses are stereotyped.
Yeah, maybe more primitive response.
I see. So in general, I mean, there's probably a lot of complexity and caveats there, but in general, right? So we're sort of born with this innate immune system. It's the part that is basically encoded in the genome. It's sort of ready to roll.
It's hardwired.
And then, so that is part of the sensing mechanism for how these gut epithelial cells in the linings of the gut are sensing the identity of the bacteria. And so what about the bacteria do we sense? Like, how does the body tell a bacteria from a human cell?
I think that the question is even more interesting than the question that you're asking now. How can this system distinguish between good bacteria and bad bacteria?
Yeah, interesting.
Because if you're thinking about these sensing receptors, they can be either on the membrane of the cells and sense basically the gut lumen, or they can be in the cytoplasm of the cells, and then they will sense something that will enter and can be recognized inside the cells. If you're thinking about recognition of bacteria by these kind of receptors, then I think we need to make a distinction between commensal bacteria and pathogenic bacteria. So if we're thinking about the gut, then we have the line of epithelial cells, and they are inside the lumen, inside our tube. There is feces and the bacteria inside it. But in between epithelial cells and the fecal matter, there is a layer of mucus. And generally, this mucus layer is preventing bacteria from getting really close to our epithelial cells. So commensal bacteria, they usually cannot penetrate and get physically very close to epithelial cells. We have this border. Pathogenic bacteria, on the other hand, they have mechanisms through which they can penetrate this mucus layer and get physically connected and physically really close to epithelial cells. So they can be sensed through physical interactions. I think that when we're thinking about commensal bacteria, the bacteria that we all have in our guts and are not necessarily considered pathogenic, then we're talking about these bugs that cannot get really close. So we need to be able to sense something that is more likely being secreted by these bacteria. So it can be secreted from bacteria and get closer and even enter intestinal epithelial cells. So when we're thinking about this, we mainly call these small molecules metabolites. So these metabolites, microbial metabolites, can be sensed as well.
I see. So I'm imagining a system here in ecology where, so you said you've got the lining of the gut, and then you've got bacteria, but they're not touching generally the lining of the gut. There's this protective mucus layer, literally physically, protecting and creating a barrier between those two populations, our cells and the bacteria cells. And if I'm hearing you correctly, the difference between a pathogen, something that's bad for us, and commensal bacteria, which is not, isn't necessarily like an intrinsic difference between them. It's simply whether or not they get too close. So could good bacteria even be bad if that mucus layer breaks down or something like that?
Yeah, exactly. So there are cases where commensal bacteria, or the bacteria that we consider good, they can become more pathogenic. And other conditions where it's many conditions that our immune system cannot recognize them and cannot maintain this balance with the antimicrobial peptides and the cytokines that are being secreted and antibodies that are being secreted into the lumen. And under these conditions where the immune system is breached, then these commensals can become pathogenic. The difference between commensals and pathogens is one of them is the location in relation to epithelial cells, but it's not the only difference. Pathogenic bacteria have all kinds of toxins and different factors that they can secrete into the cells. They have different machineries that allow them to become pathogenic and cause a disease. Commensals usually do not have these programs.
So you've got this mucus layer sort of protecting the body, not letting any of these bacteria get too close. And there are different molecules that these bacteria secrete. Some of those are sensed by the cells of the gut. Are there any general patterns as to what kinds of molecules are being sensed? Like, are these things? What kinds of things are they?
So I think it's the $1 million question. We know that in the gut there is a huge amount of these small molecules or metabolites, and the function and the identity of the majority of them is completely unknown. We don't know what exactly they are. We don't know exactly how they're being sensed. We don't know what happens downstream or after these molecules are being sensed. But over the last few years, we and others have studied few of these examples, few of these metabolites in great detail. And now we understand that some of these microbial metabolites can be sensed by different parts of the innate immune system or different immune cells. And downstream to them, many things can happen. So if short chain fatty acids that are the fermentation product of fibers that are fermented by bacteria, we cannot metabolize these kind of molecules, then they can downstream lead to secretion of different different cytokines. The function of many of these metabolites, again, is completely unknown. We found specifically during my PhD, a few of these metabolites that can activate an immune response and other metabolites that can suppress the immune response through the inflammasome complex that I've mentioned before. So some of these metabolites can activate or support an immune response, and some will dampen an immune response. But again, the majority of the metabolites is completely unknown.
I see. So things are mostly unknown here, but there's at least two very broad classes of molecules that seem to be at work here. One are molecules that we sense in order to regulate the immune response, either turn it up or turn it down. Mm-hmm. And then presumably a lot of these things are actually directly ingested as nutrients as well. Is that true?
Yeah, that's exactly true. So in addition to the regular function, or the function that we would first think of when we think about bacteria, which is processing and digestion of food, and different molecules that can be produced as a result, we can sense. So maybe let's phrase it differently. We can sense breakdown products of food that we eat. These products are either being digested by us, by our cells, or by bacteria. And we can also sense these small molecules that are indicators of some microbial activity, and they are from a microbial origin. So if we think about different, components of bacterial cell wall, or different metabolic products of bacteria that tells us, hey, now there is a very active microbial community. You need to do something about it. Maybe you need to secrete many of these antimicrobials to reduce their numbers. Maybe they're getting too close to us, to the cells. Maybe we should, produce more mucus to push them a little further away.
Mm-hmm. Yeah, I did want to ask you more about that mucus layer. It seems very important, obviously. I imagine that there are people and animals that can be put into situations where they're making too much or too little. What are there any disease states that maybe people will be familiar with that involve problems with that mucus layer?
Yeah, so one example is inflammatory bowel disease. You can have a reduced production of mucus. This enhances the inflammatory state in the gut. If there is less mucus, then bacteria can get closer, and it enhances the immune response. So basically almost any inflammatory condition in the gut can have some kind of alteration in mucus secretion. Some bacteria can degrade mucus, which makes the situation a bit more complex even.
And one of the things that's interesting here, so there's two things I want to discuss in terms of things that we put in our bodies that are probably changing the microbiome composition. One is diet and the other is antibiotics. Mm-hmm. So let's just take those one at a time. And so can you just talk a little bit, broadly speaking, about how sensitive the microbiome and its composition is to what we eat? If you shift your diet in any significant way, how sensitive is that microbiome going to be to that?
Very sensitive. So the microbiome receives many environmental signals. One of these environmental signals or lifestyle signals is diet. So when we shift our diet to a different diet, the microbes will change. The microbes see what we eat, they metabolize it, they grow or they die in response to it. So the composition definitely changes. There are many cases in which people, they travel to different countries, their microbiome shifts, but then when they will return to their country of origin, then they will go back to their lifestyle and the microbiome will gradually shift back. So we can say that genetics influences the microbiome and the composition, but it's also diet and lifestyle. And this factor, in contrast to genetics, is something that we can influence. Antibiotics deplete the microbiome. There has been a study from the Elinav lab from a couple of years ago that shows that following antibiotics treatment, if you give probiotics, it takes even longer for the original composition of the microbiome to return and to recolonize.
So say more about that. So you're saying use antibiotics, you sort of wipe out or deplete significantly the microbiome. And then people these days are often now using probiotics to replenish it. But you just said it takes even longer when you use probiotics in these cases to replenish the old population.
There are some cases where they've shown that people that took probiotics, it took them even longer to get back to their pre-antibiotics state.
Are they basically consuming the wrong antibiotic, the wrong probiotic, and putting the wrong bacteria in there?
That's a good question. So I'm not a probiotics expert, but if you think about probiotics, so I think one of the main questions is, are you actually putting something that is live into you? And whether this, bacteria that's supposed to be alive, if you bring them into your gut, into, an environment that already has bacteria in it, will they manage to survive? In the case of antibiotics, the situation is a bit different because you deplete the existing microbiome and you make an emptier niche for probiotics to grow. I'm not sure if I would, every time I would take antibiotics, I would immediately take probiotics to bring back the good bugs, because I don't think there is one recipe that fits all. My microbiome is different than yours. We don't necessarily need the same bugs. I think that something that is a bit less aesthetic is fecal microbiome transplantations, and then you recolonize your own gut with a sample of stool, basically, that was taken before you started taking antibiotics. So you bring back your own microbes. This is something that is done, in a clinical setting. Obviously not something that can be recommended to individuals at home.
I see. Now, if sometimes people take, fairly large doses of antibiotics because they get sick and they have a particular use case for it. But also, my understanding is we're probably all chronically exposed to some sort of background level of antibiotics more or less all of the time, just from it sort of being so widely used in modern society. Is there any understanding of how different our microbiome composition is because of that sort of environmental feature? Do our microbiomes resemble what they would look like if we didn't have so much use of antibiotics?
So you're talking about environmental exposure to antibiotics?
Yeah.
That is coming, for example, through the meats that we consume?
Yeah, I think it comes from the food supply. I think, people even detect it in water. Like it's sort of, there's like this background level of antibiotics sort of everywhere, it seems.
That's a good question. I'm not aware of any studies that specifically addresses this question. One way to test it, at least in animal models, would to expose them to a very, very low level of antibiotics for a really long time, and then look at their microbes. Maybe if it's the dose is too low to cause microbial killing, but it is enough in order to enhance the growth of others. And no, I will cancel this. I don't think. I'm not sure if you would be able to even answer this question.
Got it. So let's say someone takes a dose of antibiotics because they get sick. So they have a particular need to take the antibiotics. Mm-hmm. They recover. In that window where the microbiome is depleted, is there any increased susceptibility to different diseases developing in that window?
I would think that there is an increased susceptibility to other infections. So one of the main functions that our microbiome is providing is colonization resistance. So by having this huge community in our gut, whenever there is a pathogen, it's the first thing that this pathogenic bacteria would see. So they would compete on nutrients and space. And when you deplete it by using antibiotics, you basically free the niche. Now there is much higher availability of food and more space to grow. So a pathogenic bacteria can grow more. So yes, it's it enhances the susceptibility or increases the susceptibility to secondary infections.
Mm-hmm. What about, so there's this thing called the hygiene hypothesis. Can you explain what that is for people and just talk about, like, do we think that there's good evidence for that? And what do you think that means?
So yes, I think that the hygiene hypothesis is mainly linked to allergy and diseases that it's thought that we would normally not have if we would be exposed to bacteria. This theory says that in modern society and our current lifestyle, we are much cleaner, we are less exposed to bacteria, so we are more prone later on in our life to develop allergies towards different components that otherwise we would have been exposed to earlier in life and develop an protective immune response against. So I think it's definitely true. I think that in our current lifestyle, we pay much more attention to hygiene, and definitely compared to hundreds of years back. This influences the development of the immune system and susceptibilities to different diseases, including allergies later on. But I'm not an allergy expert at all.
I see. But so the basic idea is, the hygienic practices we regularly engage in the modern world, literally physically cleaning our bodies, wiping down surfaces, using disinfectants, using antibiotics, it's probably having some kind of effect on the microbiome and just the microbial world around us.
Yeah, and our immune response as a result.
I see. But you said there is good evidence that is linked to potentially the development of asthma.
Yeah, exactly.
And allergies. Interesting.
Yeah.
So what about like the subject of autoimmunity as it relates to the microbiome? So I've gotten the impression from looking at things that the microbiome is somehow connected to certain autoimmune diseases, at least. And if autoimmunity is the state where the body's basically confused about cellular identity and starts attacking its own healthy cells, how does this sort of detection and discernment mechanism of the immune system start to tie into the microbiome, and when does it start to get confused and lead to autoimmunity?
So I think the question in autoimmune diseases is, what exactly do you recognize? So if you think about the microbiome as this additional organ that you have, you can even view different inflammatory diseases like IBD as autoimmune diseases. And you can, in this case, you recognize or you have a problem in recognition of some microbial components. So this is one thing. The other thing is autoimmune diseases that are driven by different components of our cells, and then these kind of cells will be attacked by our own immune response. So we will recognize them as something foreign, while we actually need to recognize them as self and not mount an immune response against them. So in autoimmune diseases, the body is confused, or the immune system is confused, and it reacts when it shouldn't be reacting, or towards something that it shouldn't recognize as foreign.
Mm-hmm. So what you're saying is kind of interesting. So in a case like, let's just say type 1 diabetes, your immune system starts attacking some cells in your pancreas, which are important for blood sugar regulation, and they get destroyed. And the result of that is diabetes. But just like the pancreas is an organ of our body, and it literally is, it's made of cells with our genome. You're saying that one way you might think about the microbiome is it's the population of bacteria that live in our gut and elsewhere. And even though they're not us in the sense that they have a different genome, it's sort of like another organ. And we want to be unconfused about the identity of that in the context of the immune system, so that we don't attack the good bacteria, say. And so something like that can happen.
Yeah.
Interesting. So what I mean, are some of these things like IBD and some of these gut health autoimmune issues, my understanding is that they're getting more common over time, at least in the US. Is that true?
Yes, it's true. I think that this is, these kind of diseases, they are greatly influenced by our diet and lifestyle. And this is something that has changed over the last few decades dramatically. We eat different kinds of foods, we exercise less, we completely changed our lifestyles, and susceptibility to different diseases changed accordingly. If you think about higher susceptibility for cancers in the GI tract, you can, if you look at the trajectory, you can clearly see that there is a very clear increase over the last few years. I wouldn't think that, our genomes did not change so rapidly. So it's something that either the lifestyle that changed or something in our lifestyle that affects the rate of acquiring new mutations that then increases susceptibility to different intestinal cancers.
I see. So something's going on though, where in the gut, certain diseases, whether it's something inflammatory like IBD or even cancers, as you just mentioned, are going up over time. So something's going on down there. Mm-hmm. I know that one of the things you've looked at recently had to do with colon cancer and how it's influenced by certain aspects of diet. So can you describe what the basic thing was that you studied there?
Sure. So I can tell you first why we even looked at colorectal cancer. So in most of the cases of colorectal cancer, something like 2/3 of the cases, there is no known genetic mutation that is driving these kind of cancers. So we think that in contrast to a specific gene that is mutated, there are other factors, including environmental factors or lifestyle, that can make us more or less susceptible to developing colorectal cancer. And if we're thinking about specifically in the case of colorectal cancer, then one of these lifestyle factors that was associated to CRC development is diet. And in multiple studies, it was shown that different kinds of diets make us more likely to develop colorectal cancer. And these diets include diets that are very rich in sugar, Western diet, and even consumption of red meat, and specifically proteins and proteins that come from an animal source, like red meat. But from these kind of studies, we basically know what we should avoid. We don't know if there is any specific diet that will protect and not make us more likely to develop cancer. So the project that you mentioned started from this question of whether we can find a diet that will be protective from colorectal cancer or even could be used as a treatment for colorectal cancer. Yeah, so this was the beginning and the main question that we had in mind.
It's really interesting, just the idea of using diet or exploring diet as a potential treatment for certain diseases. And I want to dig into that. But why don't we start out by just describing for people, what is the diet that you looked at and what is the diet? Yeah, what's it composed of?
So we work in a lab setting, so we could design diets that were matched in their different components, and other components we could change. So we had controls and we could use diets that we specifically designed. So we designed 6 different diets, and these diets had constant protein levels, and they only varied in their fat to carbohydrates ratio, to the basically fat to sugar ratio. And what we found, so we fed mice these 6 different diets, and then we induced colorectal cancer in few different models. So we have in the lab the ability to make or to have the mice develop colorectal cancer. And these mice were fed the 6 different diets, and then we looked at the consequences. And
so,
what we
so
found,
you can just get like turn on colon cancer in certain mice.
yeah, there are ways.
Yeah, how does that work?
So there are a few ways to do it. You can induce colorectal cancer by using transgenic mice or mice that have mutations in tumor suppressor genes. And you can induce colorectal cancer by a more inflammatory mechanism, by injection of a carcinogen and then inducing inflammation. So it will be an inflammation derived cancer. So there are a few ways. They vary in their duration and slightly the outcome, the size of the tumor. So, but at least in this particular project, we used all the available models that we had. So the observation that we had could be verified across different models.
So what was so you had these 6 different diets, all protein matched. They basically only differ in the ratio of carbs or sugar to fats. Mm-hmm. And you've got these different models of colon cancer, these different types of mice that are susceptible to colon cancer. So you induce cancer in these mice, in each of the groups getting each of the different diets. And what was the basic result?
So we could follow the mice at different time points because we have in our lab a mouse endoscope. So we can perform colonoscopies on these mice and follow the progression of tumors throughout the experiment. And what we found is that the mice that were fed the diets that were very rich in fat and very low in carbohydrates, they hardly developed any tumors. And the tumors that they did develop were significantly smaller in size. If we just look at the fat content of the diets that we used, the percentage of fat varied between 0.3 all the way to 90% fat. And we found this protective effect in the diets that had 90% fat.
90.
90. Yeah. And these diets are called ketogenic diets. As I'm sure many people now know. It became a very popular diet over the last few years. And these ketogenic diets, they are very, very high in fat and they are low in carbohydrates. They have enough proteins and calories, and these kind of diets were originally developed in the 1920s as a treatment for epilepsy, and specifically in kids that didn't respond to regular medications, they were put on a ketogenic diet, and in the majority of the cases, it significantly improved the outcome in these patients.
So you find this interesting effect with tumors when you're having a ketogenic diet, 90% plus fat. Mm-hmm. You have this sort of anti-tumor effect in mice. Ketogenic diets used to treat epilepsy, so something interesting is going on. What actually? So when you're consuming a genuine ketogenic diet, how do you know that physiologically? What's going on in the metabolic processes of the body that changes?
So ketogenic diet is called ketogenic because it induces the production of ketone bodies in our body. And ketone bodies are small. They are molecules that are derived from fat, and they provide energy to tissues when glucose is not available. So when you consume ketogenic diet, the level of glucose is really low because we consume less sugars. And then different organs in our body, including the brain, they need to get their energy somewhere. So some of the tissues in our body, they cannot get enough energy by just metabolizing fat. They cannot metabolize fat, and they need to have this alternative source of energy. And our bodies, and specifically our liver, can take these fats, and by different mechanisms and oxidation processes, these fats can be converted and made into ketone bodies. And these ketone bodies can now go all the way to the brain and give the brain enough energy in these cases where glucose is not available. So these are compounds that are very energy rich, and they provide energy to these other places when glucose is not available.
I see. So when sugar's not in abundance, the body needs to get energy from somewhere. That energy is stored up in this fat, but it sounds like it's not in like the best format for all the cells of the body.
Yeah.
So the liver turns some of the fat into ketone bodies, and those ketone bodies then like go through the blood and get into the brain, and then they're used to run neurons and things like that.
Yes, we cannot function without an active brain. So these ketone bodies are a way of getting the required energy to the brain and to the periphery. It's basically an alternative source of energy.
And this state, like when you are doing this and creating these ketone bodies, that's what they call ketosis.
Exactly. Yeah, and you can get to a state of ketosis either by consuming ketogenic diet, but you can also get to a stage of ketosis when you are fasting for a long periods of times or starvation. And these ketone bodies are produced by fatty acid beta oxidation of fats that are coming from the food.
And how much does it matter what kind of fat? Like, is it any kind of fat, saturated, unsaturated, polyunsaturated? Does that stuff matter?
Yeah, so it does matter. In the case of ketogenic diets, it is recommended to consume mainly unsaturated fats and less of the good fats like avocado and olive oil. But I think that the main point here is not really what kind of fat you consume, but how much carbohydrates you bring into the system. Mm-hmm. So it's mainly the low glucose levels that drive the formation of ketone bodies.
I see. So it's a high fat diet. Maybe doesn't matter so much. What is critical is that it's low sugar, low carb.
Mm-hmm. Exactly.
And how much does like protein matter? Like, could you go into a state of ketosis if you were having a high fat, high protein diet that had low carbs?
No, I think this is the diet that is called Atkins diet. It's a different kind of diet that was designed in the 70s for weight loss. And this diet also has restriction of carbohydrates, but it doesn't restrict proteins or calories in general. So in this kind of diet, you will not go to a state of ketosis so quickly. So the main key here is to give a lot of fat, limit protein consumption, and reduce carbohydrate consumption. So if you look at the fat content in relation to carbohydrates, it's the traditional ketogenic diet is called 4:1 ratio. So you will get 90% of your calories from fat, 8% from proteins, and only 2% from carbohydrates.
I see. And so if you have too much protein, even if you're doing high fat, low carb, you won't go into ketosis. Is that because the proteins are getting used to make something other than ketone bodies?
Yes. You can break proteins to make energy. It will be a parallel process. And when you consume ketogenic diet, that has limited amount of protein. The major source of energy is fat. Will come from fat. Mm-hmm.
And how fast is this process? So let's say that you're eating a traditional Western diet today, and then you listen to this podcast and you're like, all right, tomorrow I'm going to go on the ketogenic diet. How long will it take your body to make that switch and go into ketosis?
Just a few days.
A few days.
Mm-hmm.
And do people obviously you can measure it physiologically. It has effects in the brain that maybe we'll talk about in a little bit. Do people report that they feel different?
Yeah, so personally, I haven't tried going on a ketogenic diet. I think it's a major commitment to make. But from what I saw, you deprive your body from sugar, and I think we're all to some level addicted to sugar. So I assume that the first few days are more challenging, you are more tired. I saw that people report headache and nausea and generally feeling like flu-like symptoms. But then your body gets used to it, and then you don't suffer from these symptoms anymore. And
that sounds like drug withdrawal.
I think it's really like a sugar withdrawal that we should all do.
Interesting. Okay, so anyways, let's go back to your study. So now we know what ketosis is. We know what the ketogenic diet looks like. You gave these mice this ketogenic diet, high fat, low carb, not so high protein. It had this anti-tumor effect. What else did you find? I know that there's some more detail there.
Yeah. So maybe before we go into more details in this study, I think we should mention that ketogenic diet was tried already in the past by many different groups in different kinds of cancer therapy, different kinds of tumor models. And it was shown that in some cases it is beneficial, but in other studies it was actually shown to have an opposite result. And in other cases it was shown to have absolutely no effect. So I think that ketogenic diet can be beneficial for specific cancer types, but not others. So the effect can be different. And I think that the other point that was made by other studies is that for every patient might have different mutations, even if the cancer falls under the same category. So it's not necessary that the one patient will react in a way that will be identical to another patient. So you need to take into account the different genetic alterations and tumor-associated syndromes between different individuals. But what we found in our study is that, so this ketogenic diet is protective, and we asked ourselves, how can a ketogenic diet be protective in colorectal cancer? And we already mentioned that ketogenic diet is inducing the production of ketone bodies in our liver. And these ketone bodies are beta-hydroxybutyrate, acetoacetate, and acetone. And beta-hydroxybutyrate, or BHB, is the main ketone body. And this is actually also the main source of this alternative energy. So we asked ourselves whether one of these ketone bodies is mediating the protective effect of ketogenic diet. And in order to do so, we had two parallel approaches. One is to give mice these ketone bodies. So these mice were fed a normal diet, the diet that mice are fed regularly, and they were given BHB on top of these regular diets. So they didn't go on a ketogenic diet at all.
And they just eat it? You can just consume it orally?
Yeah, you can consume it orally. You can give it in the drinking water. You can buy it in a form of a drink, in a form of different oils. It's available. It's not a drug, it's a supplement. So it's something that is very easy to obtain. And we found that it was enough to give mice BHB, this ketone body, in order to prevent and treat existing tumors.
So you recapitulated the results from before just by giving them BHB, even though they were having a regular diet with carbs in it.
Exactly. Yeah. So as long as you elevate the levels of BHB either by ketogenic diet feeding or by giving BHB exogenously from outside, you can have the same beneficial effect. We saw that so we did this kind of experiments in mice. We also used a really cool system, which basically allows us to generate mini guts, mini intestines in the lab. So we can take small pieces of the gut of a mouse or of a human patient, and under tightly regulated conditions, we can force these small pieces of tissue to form a 3D structure that resemble the intestinal epithelium. And we can treat these mini guts in culture in the lab with beta-hydroxybutyrate and see a similar response. So in mice, when we induce tumors, BHB treatment leads to protection, and the mice that are treated with BHB develop less tumors. And when we take this BHB and we give it to these small mini guts, these 3D structures, they proliferate less, so they are smaller in size. And this is also something that is, beneficial if you're thinking about cancer development. You suppress cellular proliferation.
So is BHB directly modulating some critical part of the cell cycle?
So what BHB is a molecule that is studied extensively. But what we found is that the beneficial effect of BHB is mediated by the receptor of BHB that is called GPR109A or HCAR. And after BHB binds to its receptor, something is happening inside the cell and it leads to reduced proliferation. What we identified is that downstream to this BHB, HCAR, and between this and cellular proliferation, there is another gene, and this gene is called HOPX. And this gene was previously suggested to be a tumor suppressor in different kinds of cancers. So we found that by giving BHB to mice or to intestinal organoids, these mini guts, we can induce the levels of this tumor suppressor, HOPX. And this is enough to protect. So when you treat with BHB or with ketogenic diet, if you give it to mice that don't have HOPX deficient mice, they are not protected anymore. So in order to have the protective effect, we have to have GPR109A, the receptor for BHB, and also HOPX.
I see. So BHB levels go up either because you take it as a supplement or because you go into ketosis because of the ketogenic diet or because you're fasting or something like that. The BHB is going to be sensed by receptors in cells of the gut, and then stuff happens to the genome. Genes turn on, genes turn off, and the net result of that is basically less cell division.
Exactly. Yeah, the cells proliferate less. And if you're thinking about a tumor in the gut, and the cells proliferate less, then the outcome would be smaller tumor. So the tumors will be smaller in size.
Interesting. And so, so you literally said, so BHB, it's a ketone body. It can be used to make energy. Obviously, you can give it to the mice as a supplement. Is this also a human supplement? Like, can you go to the store and buy a bottle of BHB?
Yeah, you can buy it on Amazon.
So then you just take it as a health supplement, but your study just came out. So why was it, what do people use it for already?
So people use it for different kinds of reasons. So it has this, people see it as something that is overall healthy. But BHB is used for treatment of different diseases, such as different metabolic diseases. I saw that people take it for acne, different neurological disorders such as Alzheimer's, Parkinson's.
And in the world of health supplements and health claims, generally I would be skeptical here, but I do want to talk about the effects of the ketogenic diet and BHB on epilepsy, because you mentioned it before, and I know that there's some pretty dramatic effects there. So can you summarize what that is for people and what's going on in the nervous system?
Yeah, so I think that it's not completely clear, the mechanism of action of BHB. And I think that this is something that we're lacking, especially in the field of dietary supplements. But so in the case of epilepsy, it was shown that the people got to this observation that ketogenic diet is protective, because when kids were put on some kind of starvation protocol, they found that these kids developed, or they had less seizures. And then they try to design a diet that will mimic this fasting. And they found that ketone bodies were one of the parameters in the blood of these kids that was different. It is thought that it can be beneficial in the case of epilepsy through different mechanisms. It can change the stimulation of nerves, and how the excitation of nerves, and the abundance of neurotransmitters. So it can suppress or reduce the amount of neurotransmitters. So this is one idea. And the other idea is that if you give BHB or ketogenic diet, you mimic the state that was in the developing brain. So by creating an environment that is more similar to a developing brain environment, you basically enhances or promote the recovery of neurons. I think these are the main two suggested mechanisms. One is the effect on neurotransmitters and activity of neurons, and the other one is repair.
I see. So a lot of the details are still mysterious, but BHB is having dramatic effects in the brain in terms of cellular neurophysiology. Mm-hmm. And it seems to be generally the case that when you're in a state of ketosis or BHB levels are high, that the sort of the environment of the brain is a little bit more like the developing brain and is conducive to new neural growth, which is basically the opposite of neurodegeneration.
Exactly. Yeah.
Interesting.
But I'm not a brain expert, so
What about effects on like immune responses and autoimmunity? Is there any known interaction between ketosis and that?
Yeah. So actually, when we did our study, we didn't know that, or at the point where we only had the observation that ketogenic diet is protective, we asked ourselves, how can a ketogenic diet be protective in the case of colorectal cancer? And there has been several evidence by now that it can affect the immune response by different mechanisms. One is by inhibiting one of these inflammasome complexes that is called NLRP3 inflammasome. So BHB and ketogenic diet were shown to inhibit this innate platform of the NLRP3 inflammasome. It was also shown that ketogenic diet consumption changes the composition of the microbiome, and this leads to different abundances of immune cells, and as a consequence, different immune responses. But we saw that even though we could observe similar results in our system, so also when we feed mice ketogenic diet, they have less inflammatory cytokines. So the NLRP3 inflammasome is important, but it doesn't mediate the protection effect in the case of colorectal cancer. So if you take NLRP3 knockout mice and wild type mice, and you treat both of them with ketogenic diet, and you look at the cancer development, the knockout mice and the wild type mice will both be protected when they are fed ketogenic diet. So NLRP3 might be affected by ketogenic diet, but it's not driving the protection in the case of colorectal cancer. It has many different and other important functions, but not in mediating a protection from CRC in mice. The same is true for T and B cells. We used mice that don't have any of these adaptive immune cells, and also their ketogenic diet was protective. So even though these mice are deficient in a very major part of their immune response or immune system, then even though they don't have this part, they can still have the protection that is mediated by ketogenic diet or beta-hydroxybutyrate. Mm. So ketogenic diet can change many different aspects of our physiology, but it's not all these different parameters do not necessarily drive the protection.
Mm-hmm. So far the ketogenic diet is very popular right now. I mean, a lot of people have at least heard of it. A lot of people have tried it. So far in our conversation, we've talked about only good things, right? Like ketogenic diet can have this anti-cancer effect sometimes, although sometimes you did say it is possible to have the opposite.
Yeah.
It has this anti-epilepsy effect. Are there any downsides here? Is there any reason, everyone shouldn't just go buy a bottle of BHB and start consuming it?
So I think we should make the distinction between ketogenic diet and BHB. So I think that ketogenic diet was shown to be beneficial or not beneficial, depending on the type of cancer and the context. It was even shown to enhance the efficiency of other kind of cancer therapies. It was shown that ketogenic diet can enhance the efficacy of targeted therapies. If I remember correctly, PI3 kinase inhibitors, if you give ketogenic diet when you treat with these kind of inhibitors, you make the treatment more effective. It improves the efficacy of the classic cancer therapies in some cases. These kind of diets, they are very, very rich in fat. So you would think that they will induce elevated cholesterol or, negative consequences on our general metabolic health. But it was actually shown that it doesn't greatly change the body composition or the different parameters in our blood. And as far as I know, no studies reported any severe adverse effects of ketogenic diets. So it's, as far as I'm aware, it doesn't cause any deterioration of metabolic markers, not cholesterol, not triglycerides. Yeah.
And what about BHB supplementation? Is there any reason to be cautious about that?
I'm not a physician, so I cannot say if there are certain conditions where BHB should not be taken. I think that if you compare just BHB supplement and you compare it to a whole ketogenic diet, I think that in terms of how practical it is, I think that drinking a BHB supplement is much easier than adhering to this very, very strict diet that doesn't have any carbs and basically limits. It's almost an anti-social kind of diet because you cannot go out, you cannot have alcohol, you cannot, So I think that just drinking BHB is something that is more realistic. I'm not aware of any negative side effects of consumption of BHB or these ketone supplements. I saw that some of these supplements, they can cause some discomfort in the gastrointestinal tract, but this is something that usually goes away, I think, after a few days. Not all supplements taste good, but other than this, I'm not aware of any major side effects.
Yeah, and I would imagine too, there's feedback here. Like if you go into a state of ketosis for a prolonged period of time, is that so if you were to take if you gave mice or humans, but maybe you've done this in mice, if you give them BHB supplementation but not the full ketogenic diet, so their diet doesn't change except that they've got this BHB supplementation, is that sufficient to cause clear changes in the microbiome composition?
So ketogenic diet definitely alters microbial composition. In the case of BHB, it's less clear. I'm not sure we even looked at it, Yes, but I would think that yes, just because BHB can change our innate immune system, which then can modify and affect the composition of our microbiome. So my guess is yes, it will change.
The other thing I want to ask about is, so obviously our bodies have the capability of switching into these different metabolic states, ketosis, and then, I'm sure there's multiple other ones that I don't even know about, but the fact is we can do it. And if you think about, think about this in evolutionary terms, if you think about wild animals or human hunter-gatherers, present day or in the past, they probably generally lived in environments that were much less stable and plentiful than ours in terms of food availability. So they had to go through periods of feast and famine. Sometimes food was abundant, sometimes it was scarce, and you didn't have a choice as to whether or not you were going to be fasting. It was just sort of a part of the natural cycles of life. So, I mean, with that in mind, when we think about sort of the natural human ecology that our bodies evolved under, are our bodies sort of like expecting to switch metabolic states at different times and not always be in sort of like one diet, one mode of metabolism?
Yes, I think, it's exactly what you're saying. Our body has to have this backup in case we will encounter a long periods of starvation, or when food will be less available to us. We can lose weight, but our brains, our brain has to have enough energy in order to continue function. And it's not just our brain, it's other organs like the heart and our muscles. Not all these peripheral organs can get enough energy in the absence of glucose just by consumption of fat. They rely on these ketone bodies to get to them and to allow them to get enough energy. Yes, it's definitely, it's something that is very important from an evolutionary point of view, because in the past, food availability was much less predictable.
What are some of the big questions that you guys are working on in the lab now?
So I think that at least in the case of the ketogenic diet or BHB story, there are few next interesting questions. One is whether the microbiome is playing any role in this protection. This is something that we're currently addressing, so I cannot comment about it at the moment, hopefully soon. The other question is whether you can combine BHB with existing treatments for different kinds of cancer. So we identified one specific mechanism by which ketogenic diet is protective in the case of colorectal cancer. I think that it will be interesting to get to this kind of mechanistic understanding of the effect of ketogenic diet or beta-hydroxybutyrate in other cancers. So most of the work that we have done and that we are currently working on in my lab is based on animal models of cancer. We have some evidence that is also included in the paper that was recently published that suggests that a similar phenomenon can also be observed in humans. So we found that in colorectal cancer patients, the amount of BHB correlates with these different genes that we identified, specifically HOPX. So when patients have higher BHB, they have higher levels of HOPX, and they also have reduced cellular proliferation. We found it by single cell RNA sequencing. And yeah.
Oh, I was just going to say this. Yeah, I mean, this is a really interesting finding just beyond your specific results for colon cancer and the ketogenic diet, because it's sort of, at least to me, it's saying, well, if one of the main cellular physiological effects here is to limit cell proliferation, it's very natural to suspect that this isn't going to be limited to the colon. And it also starts to get you thinking about, like, okay, if the Western diet and the Western lifestyle is having this sort of pro-cell proliferation effect, it starts to point to a story where, like, yeah, maybe that's exactly why a number of cancers are on the rise and associated with this kind of general lifestyle.
Yeah, I think that's, so we identified this specific pathway of ketogenic diet, BHB, and how it acts inside the cell. I wonder if, in order to observe a similar protection in other cancers, or even in other organs, or in different cancers along the GI tract, would you need to have the same cellular machinery in order to have the protection? Or let's say you will now observe a protection in cancer in a completely different organ. Do you need to have the same receptor, the same protective genes in a way in order to have protection? Or are other genes important in different cells?
Mm-hmm.
So I think that the mechanism can be different between cell types, between different types of cancer. This is a really interesting question. We currently are in the process of starting a clinical trial in colorectal cancer patients. So we will treat patients with BHB, and we will see if we can observe something similar, if we see this protection also in humans that are treated with BHB and not just in mice. So this is something that is currently an active ongoing clinical trial that hopefully we can get the first results from in the next couple of months.
How much BHB do you give to patients?
So we give the recommended amount. The patients will drink BHB drink 3 times a day, and it's a relatively small volume of approximately 30 mLs, 30 milliliters.
Yeah, so it's not like a mega dose or something. It's a normal amount. Yeah.
Yeah. Yes.
Interesting. Well, this is fascinating stuff. I mean, I think this is an interesting area, just the general idea that diet can have such a physiologically potent effect on us at the level of, preventing disease and things that maybe we haven't appreciated historically as much as we are starting to. So I'm definitely looking forward to seeing what else comes out of this work from your lab.
Thanks. I think it's really exciting, and I think that it will be really interesting to see if we can design diets that will be protective or prevent different kinds of cancers, different kinds of diseases, understanding, what is the mechanism of protection in case we identify such, and whether we can design diets that will support existing therapies. The therapies, at least in the case of colorectal cancer these days, have major side effects, chemotherapy and surgery. If you could maybe, give patients BHB and, reduce the chemotherapy or, change the amount of chemotherapy they would need to take, you would improve their quality of life significantly. So I think there is a lot of potential of treating with BHB or even other molecules that can be identified that are coming from diet or that are influenced by diet, either as a standalone therapies or in combination of existing standard therapies. And it's not limited just to BHB. It can, the same kind of studies can be performed with different diseases, different diets, different molecules, BHB, different microbial metabolites. I think the possibilities here are endless.
Well, I think that's a great spot to end it. Dr. Maayan Levy, thank you for your time.
Thank you so much. My pleasure.